Fluoppi technology. To begin with the introduction of Fluoppi technology, let's take a look at the movie first and learn what Fluoppi technology is like. Fluoppi can visualize the presence of PPI by forming fluorescent punctum. In this movie, mTOR and FKBP12 interaction is induced by addition of rapamycin. Now let's see the movie. The time scale shown in the bottom is minute and second. As you can see, fluorescent punctum are gradually formed after exposed to rapamycin. Then how this technology works? This slide is a schematic representation of Fluoppi. Two tags are used for Fluoppi. One is a tetrameric fluorescent protein, FP tag. Another is an ash tag, which forms tetra to octamer. Protein X and Y is fused to these tags, respectively, and then transfected into the cells. Before X and Y interact, these molecules are diffusely distributed. If X and Y interact, they form clusters using their multiple arms. Because X has fluorescence, we can visualize intracellular PPIs as fluorescent puncta. If the multimeric tags are substituted for monomeric version of tags, fluorescent puncta do not form. This is an example of mTOR and FKBP12 interaction. As you can see, fluorescent puncta were not detected even after addition of rapamycin, if monomeric tags are used instead of multimeric tags, which means both multimeric tags are required for puncta formation. Formation of puncta is reversible. This is an example of p53 and MDM2 interaction inhibited by NUT23. p53 peptide and partial region of MDM2 was fused to ash tag and azamigrin respectively. An engineered cell line expressing the construct was developed from CHOK1 cell line. Before adding NUT23, all of the cells has fluorescent punctum. However, after addition of the compound, they immediately dissociated in several minutes, which means NUT23 was entered into the cell within several minutes. In this experiment, we kept monitoring the p53 and MDM2 interaction after washing out the once treated NUT23 compound. Let's see the movie at first. As you can see, fluorescent puncta were recovered after washing out the compound. In this chart, x-axis represents the time and y-axis is the fluorescence intensity of puncta region. So with Fluoppi technology, you can visualize intracellular residual time of your compound. Using Fluoppi, MBL has visualized 30 or more different PPIs. The results are indicated here by protein length and reported KD value. X and Y axis show the length of PIP proteins, and the plot side shows KD value of the PPI in publications. Gray dot indicate their KD value have not determined. As you can see, Fluoppi is compatible with 10 to 2000 amino acid length of proteins and a wide range of KD values in living cells. We compared Fluoppi to another technologies for analyzing intracellular PPIs. We classified those technologies into three groups. The first one is redistribution principle, which is employed by Fluoppi. The second one is detecting proximity of tags fused to target proteins, like FRET or split GFP. The last one is the reporter gene assay. Comparing Fluoppi to those technologies written here, Fluoppi can monitor both on and off of PPIs in real-time scale. And very distinct punctum-based fluorescence signal provide very strong intensity and high signal-to-background ratio. Because Fluoppi is an imaging-based technology, a lot of information, including cell-to-cell viability, can be obtained in addition to the presence of PPIs. And also, setup of Fluoppi is easy, as the construct do not require optimization of linker lengths. Hence, Fluoppi is a simple and robust system for PPI detection and screening of PPI modulators. In summary, Fluoppi visualizes intracellular PPIs as fluorescent puncta. Wide variety of PPIs can be applied for Fluoppi, and also reversible reactions can be visualized in real-time manner. Because Fluoppi has a unique mechanism of action, it can be a technology to complement the existing ones.